Hi. As a part of the workshop and with respect to the assignment 2, so I have been given task of submitting the research review of the paper which was given to me. So the title of the paper highlight is that level-free plasma proteomics identifies I hepatoglobin-related proteins as a candidate marker of idiopathic pulmonary fibrosis and dysregulation of complement and oxidative pathway. And my so with respect to this paper, the one of the co-author of this paper is Mayank Shashwat. A few detail about him. He is basically presently in the Stanford Brumman Pre-Bice Medical Discovery Institute, and he is expert in the field of gynecology, molecular biology, biochemistry, mass spectrometry, and glycoproteins. He has around 26 number of paper published, and the citation scores for this for all of his articles is 1234 since 2019. These are the few details about the corresponding author. With respect to the abstract, I'll be highlighting few points from the abstracts that so this abstract highlights the use of level-free plasma proteomics to identify potential biomarker for idiopathic pulmonary fibrosis, are called IPF, a progressive lung disease with poor diagnosis or prognosis. It points toward the dysregulation of complement activation and oxidative pathway in IPF patient with heptoglobin related proteins identified as a candidate marker for the disease. Coming to the introduction part, IPF or idiopathic pulmonary fibrosis is a progressive lung disease with a poor prognosis, primarily affecting older adults and lacking specific diagnostic biomarkers. The current diagnostic method for IPF rely on clinical and radiological assessments, leading to delayed diagnosis which can impact patients' outcome. The study, this study aimed to provide address to the critical need for novel biomarker in IPF by conducting level-free plasma proteomics on samples from IPF patient and healthy controls. Uh, Through proteomics analysis, the research identified hepatoglobin-related proteins as a potential biomarker for IPF and highlight dysregulation in complement activation and oxidative pathway in IPF patients. Some of the key findings of this paper is hepatoglobin-related proteins as a potential biomarker. The studies identified hepatoglobin-related proteins as a potential biomarker for IPF, showing promise for early diagnosis and disease monitoring. Second, dysregulation of key pathway. The research also revealed that dysregulation in complement and oxidative pathway in IPA patient, offering insight into the disease mechanism and potential target for future treatment. Essentially, these studies contribute valuable knowledge in the field by suggesting a new biomarker for IPF and highlights potential pathway involved in the disease. The methodology session or method sessions, I would like to highlight few points like the studies recruit IPF. In this study, it has been recruited IPF patient from the Finnish IPF registry and healthy controls from Helsinki Biobank, ensuring age, sex, and smoking status matching. Level-free plasma proteomics were performed in 36 samples, including 17 IPF cases and 19 controls, following the 2011 guidelines for the sample collections. Proteomics data analysis involve volcano plot, hierarchy clustering, partial least square discrimination analysis are called PLSDA, and ingenuity pathway analysis to identify potential biomarker and pathway associated with IPF. Univariate and multivariate statistical analysis were employed to access protein difference between IPF and control sample with subsequent evaluation using rock curve analysis. Pathway analysis revealed the enrichment of pathway related to LXR, RXR activation, complement system, and coagulation cascade in plasma protein of IPF patient. The results it the result discussed in this paper are successful differentiation between the IPF patient and healthy control. The research were able to differentiate between the blood of the blood plasma of IPF patient and the healthy controls with good accuracy using proteomic analysis and statistical modeling. This suggests that the identified protein difference are significant and potentially used for diagnostic purposes. Hepatoglobin related proteins as a potential biomarker. Second point, this study identified HPR as significant less abundant in the blood plasma of IPF patient compared to healthy controls. And this makes HPR a promising candidate for novel biomarker for IPF. Then dysregulation of complement and coagulation cascade. The, the research found that the protein involved in the complement and coagulation cascade were significantly different in IPF patients. This suggests that the pathway are dysregulated in IPF and could be potential target for future therapies. Other dysregulation pathways, beside complement and coagulation pathways, the study also identified dysregulation pathway in other pathway related to inflammation, lipid metabolism, and cell signaling, especially the ERK1 or ERK2 pathway in IPF patient. The conclusion drawn from this paper are HPR as a potential biomarker, as the study identified hepatoglobin-related protein as a potential biomarker for idiopathic pulmonary fibrosis. This is significant finding as HPR showed a considerable decrease in IPF patient compared to the healthy individual. Dysregulation of complement and oxidative pathway. The researcher also confirmed the involvement of complement and oxidative pathway in IPF development and progression. This reinforce provide finding and highlight this pathway as a potential target for future therapeutic intervention. Future research needed. While study present a promising results, the author acknowledged the need for further validation of their finding in larger and more diverse patient cohorts. This is crucial to confirm the reliability of HPR as a biomarker and to explore the identified pathway for developing effective treatments. In essence, the paper also concludes that HPR holds as a potential biomarker for IPF, and the dysregulation of complements and oxidative pathway play a crucial role in the disease pathogenesis. However, future research is necessary to solidify this finding and to translate them into the clinical applications. Future work suggested in this paper are further mechanistically studies are needed to establish the dysregulation system pathway in idiopathic pulmonary fibrosis, that is IPF, for better understanding of the disease and to guide future research in diagnosis and treatment. Future studies should focus on additional validation of hepatoglobin related protein HPR in larger independent cohorts to confirm its potential as a biomarker for IPF. The potential of HPR as a monitoring biomarker should be explored in future studies to assess its level in response to the treatment, aiming to restore them towards healthy levels in IPF patients. So few of the limitations of this paper are the studies also had the modest number of IPF samples, which may be limit the general which may relate to the general liability of the finding to a larger populations. Lack of highly specific antibodies against hepatoglobin related protein HPR may pose challenges for future validation studies and potentially requiring mass spectrometry based assays. The research acknowledged the needs for validation in more numerous cohorts of IPS patients and other intestinal interstitial lung diseases to confirm the identified biomarkers and the pathway. This the study was designed as a binary case control comparison, which may not compare the full complexity of IPF pathogenesis and their progression. Significance of this paper. IPF is a serious condition as it is chronic and progressive lung disease with poor prognosis. Early detection is crucial for better management and treatment. Limited biomarker. Second one is limited biomarker. Currently, there are few reliable biomarker for IPF, making a diagnosis and monitoring challenges. Third, for potential for new markers. The studies uses a proteomics to identify potential new marker in blood plasma, which is less invasive method than lung biopsies. Fourth, hepatoglobin related protein. The studies identified hepatoglobin related proteins as a promising candidate marker for IPF. This could lead to early this could lead to a earlier diagnosis and better disease monitoring. Insights into this mechanism, the researcher also sheds light on dysregulation of complement and oxidative pathway in IPF, potentially opening avenue for the new treatment strategies. How are this research going to benefit us? The present the research presented hold a significant potential benefit for the future idiopathic pulmonary fibrosis diagnosis and treatment. So first point, early diagnosis. The identification of HPR as a potential biomarker could lead to the early diagnosis of IPF. And this is crucial because early intervention is associated with better outcome for progressive diseases. A simple blood test measuring HPR level could potentially detect IPF at an early stage than a current method. Second, monitoring disease progression. Monitoring HPR level over time might also help doctor to track the progression of IPF in patient, and this could allow for more personalized treatment strategy and better management of the disease. Third, new treatment target. The confirmation of dysregulation complement and oxidative pathway in IPF offer valuable insights into the disease mechanism, and this knowledge could pave the way for developing new drugs and therapies that target this pathway and potentially slowing down or even halting the disease progression. Last point, improved quality of life. Ultimately, the finding of this research have potential to improve the quality of life for IPF patient. Early diagnosis, better monitoring, and more effective treatment could all contribute to longer survival time and a better overall prognosis for individual living with the challenge with this challenging disease. Thank you. That is the quick short presentation from my side, and thank you for giving the chance.